All right, looks like we are live and we've got a great audience and a great chat already. Some great people in the live chat. Thanks for being here. This is definitely going to be a debate to remember. I'm extremely excited for this one. And real quick, actually, before I bring in Jackson Rowe, as well as Praise I Am, who is going to be moderating this debate, I want to go over the topic as well as the format for the evening. So we are going to be debating human evolution versus independent origins, created or evolved. Now, the format for the night is pretty similar to the debate I had with Bill Tavis last week, which was definitely one to remember. We are going to be having 14-minute opening statements. I should say up to 14. So 14 will be the max that we can take for our openings, and whatever we don't take, we can just throw into the audience Q&A, followed by 10-minute uninterrupted rebuttals, followed by another rebuttal round of 5 minutes, and then a 25-minute open discussion where we can ask each other questions pertaining to the topic of the debate, ending with 2-minute rebuttals to close out the discussion and a 5-minute closing statement. And then, of course, roughly a 25-minute Q&A with the audience. So please, everybody in the chat, make sure you are tagging Praise I Am and not myself, because I won't see it. I will be debating. I know you guys are probably used to me moderating, but today I'm debating, and that is why we've got Praise here, the #1 moderator, as well as Jackson Rowe. Gentlemen, thanks so much for being here. Let me unmute you guys. Praise, thanks for being here for moderating, and Jackson, thanks for being here for this important debate.
Thanks for having me.
Looking forward to it. Hopefully we'll keep this a cordial, respectful, and entertaining debate tonight.
Awesome. I'm pumped. Yeah. Well, why don't we, since I just went over the format, why don't we hand it over to Jackson Rowe for a brief introduction to kind of break the ice. Jackson, who you are, what's going on over at your channel. For example, if people were to go check it out, what are they going to find, and so on and so forth. So thanks for being here, Jackson, and go ahead. The floor is yours for just a brief introduction as to who you are.
All right. Well, thanks for having me. Yeah, my channel's called Jackson Rowe Science Show, which is kind of like Bill and I, the science guy. That's why I chose it. It kind of rhymes. And I started like probably just a month ago. Doesn't have many subscribers, but I think I've got some decent content. Not great production value, and I'm kind of monotone, so I don't have that going for me either. But yeah, I live in Arkansas. I'm 27 years old, and I've been into science since I was a little kid. So that's really about, well, I'm into animals. I have a lot of exotic pets. That's about it. If you want to, they probably know all about you, but if you want to talk about your intro.
I appreciate that. Yeah, real quick, I'm definitely no stranger to this channel since, I do run the channel. So I'm standing for truth. I've had roughly between 80 to 100 debates. I've been doing this for almost 3 years. It's been a lot of fun. It's been quite the adventure. This is definitely one of my favorite topics to debate. I've debated all sorts of topics, but I like this one because it focuses on the bigger picture. You know, whether we're created or evolved, independent origins, human evolution. If independent origins is true, then obviously that kind of takes out abiogenesis and also takes out universal ancestry with it. So it's a good debate. It's an exciting topic because we're focusing on the bigger picture. So I've also written a bunch of books on this topic. A number of them are behind me. So anybody wants to check those out, I've written about 5, as well as co-authored a large number as well with Ramat. So he also runs this channel with me. So that's about it from me. And looks like we got a super sticker flying in as well from Jeremy Martin. Thank you so much, Jeremy. I appreciate it. And we've got a good chat. So introductions are out of the way. Jackson, thanks for doing this debate. I'm excited. And we're going to hand it over to you. Okay. Praise. Looks like we are going into our 14-minute opening statement. So Praise, how do you want to work it? Are you going to give us a 1-minute warning, or? Oh, Praise, you're on mute, brother. There.
Yeah. So, yeah, how I'm going to do this is when you guys get near the end, I'm going to say, go ahead and wrap it up. I'm going to give you about a minute to finish up and conclude your statement, and then we're going to move on to the next interlocutor.
All right. All right. So I'm going
That's
to jump
why
in.
Yes. Thank you, Praise.
All right.
And Jackson, whenever you're ready, let us
Okay.
know, and Praise will start your timer.
All right, you can go ahead. I timed it out. It's about 10 minutes probably, so I probably won't need all the time. All right, my position is that humans evolved from previous forms of primates. Humans and other great apes share a common ancestor. I'll present fossil, genetic, anatomical, and behavioral evidence to that effect. I believe that this evidence doesn't necessarily preclude a God, even the Christian God. Millions of Christians accept evolution, including most Catholics and Methodists. In fact, the majority of people who accept evolution are religious, by one poll I saw anyway. While I personally don't believe in a God, I don't think modern science and God are completely incompatible. But back to the subject matter, I'll be presenting evidence about Australopithecine bipedalism, which I saw you give a talk, or Dr. Biddle give a talk on that on your channel. Modern ape anatomical, genetic, and behavioral similarities to humans, and probably some miscellaneous stuff thrown in. So first, I'll just touch on that, the Australopithecine stuff. Most creationists seem to only be aware of the Lucy fossil, which is Afarensis, but there are actually many species and many fossil specimens, not only of other species, but of Afarensis as well, including the what's called the big man fossil, which shows a partial pelvis as well, and the Salem child, which has most of its spine and skull. There are also about 400 individual fossils of Afarensis, though most of those are just singular bones or a couple of bones. But from those bones, we can tell that, for example, the Afarensis foot was built for bipedalism. Its heel was robust, unlike a chimpanzee and more like a human. Its fourth metatarsal, which has been found, indicates there was a stiff midfoot, unlike a chimp, and also an arch, like a human, an adaptation for walking. Same thing with the Africanus foot, Australopithecus africanus. Now, the Australopithecine spine was more S-shaped than the chimp spine, which is more arch-shaped. That's an adaptation for bipedalism. They can tell that from different spacings and different components of the vertebra. So also the foramen magnum, which is the hole in the underside of the skull, is intermediate between a chimp-like ape and a human-like ape. So it likely held its head similar to ours as it was upright. Its femur is angled the same way as ours, as opposed to a chimp's. And the pelvis has been kind of contentious with Lucy. I know some people have argued that it was filed down or faked, but there have been several Australopithecine pelvises found, including 2 Sediba pelvises, 2 Africanus pelvises, and 2 Afarensis pelvises, including Lucy's. Also Prometheus, which may actually just be an early Africanus, but some say it should be its own species. Now, where was I? The ankle joint of the Australopithecines are square-shaped, the ankle bone, where chimp is trapezoid-shaped, which allows for more flexibility for climbing, which the Australopithecines apparently didn't have, except maybe for Sediba. The rib cage was originally thought to be more cone-shaped, like a chimp, when Lucy was first discovered, but further finds have indicated that it's actually more barrel-shaped, like ours, so they would have had more of a waist than great apes have. The previously mentioned Prometheus species, which is known from the Littlefoot fossil, which is the most complete ever found of an Australopithecine, shows that the arms were in fact shorter than the legs, which is unlike gorillas, orangutans, chimpanzees, and a very human-like feature. The brain capacity was about 450 cc's, where a chimp's about 400, and early Homo species are in the 500 range, so it's intermediate between there. I'm not saying we evolved from chimpanzees, I'm saying we share a common ancestor. I'm just using chimps as an example. In fact, the line between Australopithecus and Homo isn't so clear-cut. Homo habilis, there's some debate about whether it should be classified as Australopithecus or not. Most scientists say it should be Homo as it is. There are a few that say it should be Australopithecus. And even Homo erectus, in its earlier stages, where it's called Homo ergaster, which is no longer recognized as a species, but actually just an early Homo erectus, had a very small brain capacity, had some more primitive traits. Same with Homo georgicus, which is also just an early Homo erectus. And Homo erectus is the most known from the most complete hominid skeleton ever found, the Turkana boy. I forget when that was found. I should have written that down. But anyway, the Neanderthal genome has been sequenced, and it shows that creationists often argue that Neanderthals were the same species as us, Homo sapiens, but it shows that we differ by 202 base pairs, where by comparison chimps differ by 1,400 base pairs. So they were very close to us, but they weren't quite human. Now moving on to modern great apes. While it's true that 99% of the regions that code in our DNA and chimps are the same, it's been argued that it's not 99% all the way around, which is true, but it's not dishonest to say that they're the closest living relatives we have. No other animal, plant, fungi genome comes close to ours the way a chimps does. In fact, chimps are more closely related to humans than gorillas are to chimps. And molecular clock data, which is mutation rate between generations, indicates that our common ancestors split off from chimpanzees 6 to 13 million years ago. Now, one of the best evidences for common descent in the genome is are the endogenous retroviruses, which is when a virus infects the gamete cell and gets passed on to the future generation. And that's important because we share 99% of ERVs with chimpanzees. And that could really only be true if we shared a common ancestor with them. Now, behaviorally, there are some real similarities. Kanzi the bonobo has been taught to use hundreds of lexigram symbols to form novel sentences and understand novel sentences. And while that's not unique in the animal kingdom, it seems unlikely that something that wasn't our closest relative would know. Same for Coco the gorilla. She knew 1,000 sign language signs, understood thousands of spoken words, displayed human-like emotion. Chimpanzees are the only other animal known to have ever fought in an actual war, the Gombe Chimpanzee War, which took place in the early 70s. Over the course of 4 years, one tribe of chimpanzees totally wiped out the other tribe. So it's just a unique thing among the animal kingdom that we share with chimpanzees. Anatomically speaking, we should have the same bone number as the other great apes. We have the same number of teeth. We have the same teeth layout, the same number of incisors, canines, premolars, and molars. We have the same trichromatic vision. We have the same ABO blood type system. We have dermal ridges on our fingers, which is very rare in the animal kingdom. I think only the koala has those, other than the great apes. I may be wrong on that, but also our fingernails are very similar and unique in the animal kingdom. Now, some arguments against the Adam and Eve argument. primarily inbreeding. Now, even if Adam and Eve had the perfect genome, as has been suggested, after the fall, they would have started to acquire mutations from just mutagenic factors like UV radiation, and they would have passed that on to their children, and then their children would have acquired more mutations through the same factors, and that would have been amplified when the children had to inevitably reproduce with each other. And from there, it gets just really, really bad. They just more inbreeding brings recessive traits, harmful mutations to the surface, and pretty quick you get all sorts of negative mutations that are passed on and not selected out. There's a good example in the animal world, the animal breeder world, like ball pythons, axolotls. When you inbreed those, you get these color morphs: albino, leucistic, piebald. It's hard for me to read my handwriting. Hang on. Why were they even given the option to eat the apple? I think it's because the story is an allegory. Why were they made from dirt and then a rib? I find that hard to believe, and again, it goes back to being allegorical. And also the talking snake speaks of allegory as well. And why would God, being omniscient, not be able to see the future of Adam and Eve eating the apple in the first place? You would think he would, being omniscient, see that outcome. And why would all humans be punished for the mistake of two? The concept of original sin doesn't really make any sense to me, but that's kind of a different debate. Also, mitochondrial DNA doesn't really support the claim of Adam and Eve. Y chromosome Adam and mitochondrial Eve didn't live at the same time. Mitochondrial Eve, our last female common ancestor, lived about over 100,000 years ago, and evidently Y chromosome Adam lived tens of thousands of years apart. And nuclear DNA shows that the population of humans was never below about 10,000. So that kind of precludes the idea of only two human ancestors. That's pretty much my opening statement. I'm not sure I did it right. It's my first debate, but not sure how much time I used. But standing for truth, if you want to take it from there.
So you had about a minute and a half to spare, so we can add that in the question and answer period, or if you want to add that to your finishing comment later on. So SFT, you're next in for your opening statement, and whenever you're ready, then I'll start the timer.
Okay, awesome. Praise. I'm going to share screen right now, and then let me know if you can see it. Just 1 second.
Yes, I see it.
And then I'm just going to mute you, Praise, because I hear like a little beeping noise, which is fine. So just when you give me that 1 minute warning or 2 minute warning, just make sure you unmute yourself for that. And I'll let you know when to start the timer. Just 1 second. All right, Praise. As long as the audio video is good, maybe I'll get the A-OK from the audience as well. We can. Audio good, video good, or audio good, slides are showing?
Yeah, I can see it.
Awesome. Okay, here we go. So, Praise, you can start the timer right now. Tonight's debate is human evolution versus independent origins. Are humans specially created and made in the image of God? Or did humans evolve slowly over time from ape-like ancestors? Are humans related to chimpanzees and the other great apes? I love this topic because the evidence for independent origins also demolishes the so-called evidence for human evolution. I am going to waste no time and just get right into the data. My opening will actually rebut many of Jackson's arguments that he put forth in his opening statement, and then I'll save the rest for my rebuttal round. So here we go. To start, we have the major unsolvable problem of mutation accumulation and genetic degeneration. Mutations are accumulating far too quickly for natural selection to filter out the damage. What type of selection can Jackson invoke that will counterbalance the accumulating damage? DNA is a very fragile molecule. From one generation to the next, many mutations are passed on. Roughly 100 new mutations per person per generation. We are consistently more mutant than our parents. As a matter of fact, we are all multiply mutant, which is why selection is so limited. This is 100 typographical errors every generation that our parents did not have. Natural selection must remove or filter out these mutations, or else we will go extinct. The problem for Jackson here is that these nearly neutral mutations are only slightly deleterious and only slightly harmful to the point where they do not really affect reproduction. Selection cannot see these, which means species are going downhill. And Jackson does not have millions of years of evolutionary advancement. Low impact deleterious mutation accumulation puts shelf lives on genomes, meaning Jackson does not have the deep time he needs to evolve an ape-like creature into a human. We also see a perfect biological decay curve that matches the biblical model of longer lives closer to the creation event, while it drops off after the flood. This data has almost no chance at being coincidence. This is an amazing correlation between what we know about mutation accumulation, genetic degeneration, and biblical longevity. It makes no sense that the biblical authors would ever make this up. Also, fabricating this biological decay curve would require exceptional knowledge in statistics and mathematics. The systematic degeneration of man as documented in the Bible is extremely consistent with genetic entropy and deleterious mutation accumulation. The Genesis biological decay curve reflects real biology. Also, many of the so-called icons of human evolution are actually not evidence for ape to man evolution. They are evidence for genetic degeneration in the past. I will go over just a couple of these for now, and this also addresses some of the icons that Jackson brought up in his opening statement. So we've got evidence that Homo neanderthalensis was highly inbred, had a very high genetic load, 40% less fit than modern humans. This severe genetic degeneration probably contributed to the disappearance of that population. Also, the hobbits, Homo floresiensis, was also inbred and apparently suffered from a special type of genetic degeneration called reductive evolution. This results in reduced body size, reduced brain volume, and various pathologies. The hobbit-like human species may turn out to have simply been the remains of a human suffering from genetic illnesses. And this is exactly what we would expect given the post-Babel model, where you've got these people groups isolating themselves after migrating from Babel. What you get is founder effects and rapid genetic degeneration due to inbreeding. The anomalous features that the evolutionists like Jackson like to point out are transitional are actually due to pathology and degeneration. Homo naledi, same thing, likewise inbred and suffered from reductive evolution. Results in the same types of anomalous features: reduced body size, reduced brain volume, and various pathologies. Homo erectus, which Jackson brought up, same issues. Was inbred, suffered from reductive evolution, all consistent with the post-Babel model of isolation and inbreeding. So let's get into the next portion of this opening here. Jackson also talked about molecular clocks. So based on the Genesis creation account, we can make some extremely specific predictions and retrodictions. These have all been confirmed. We see low genetic diversity in humans, which is a direct expectation if we really did originate from just two people thousands of years ago. Starting from just two people automatically restricts genetic diversity. If we have been evolving for millions of years, we would have populations with a lot of mutations floating around, which would suggest we have high levels of genetic diversity today. We do not see this. We also have strong evidence for one mitochondrial DNA ancestor and one Y chromosomal ancestor of all people on the planet. The mitochondrial DNA compartment that we only get from our mother picks up a mutation about every other generation. The mitochondrial DNA mutates far faster than evolutionists have ever predicted. When we build these phylogenetic trees or family trees, we can look at mitochondrial DNA diversity worldwide. We can also see how far apart people are. And with the fast mutation rate, Eve, the mother of us all, existed only a few thousand years ago. There are only a few DNA differences separating any two people on the planet. How does Jackson deal with this data? Paper after paper confirming this. Oh, just one second here. The Y chromosome as well is also fast, picks up about one mutation every generation. There are only a few hundred mutations separating people worldwide. And our last Y chromosomal ancestor, being Noah, existed just 4,500 years ago. This is all inconsistent with universal common ancestry. I want to go to a phylogenetic tree here because I've pointed this out in the past. Dr. Robert Carter, he took all the Y chromosomes and made a Y chromosome family tree. This tree is incredible because what we see is exactly what we would expect if independent origins and biblical creation were true. What we see visually is an explosion from a central point. This is an explosion essentially from Noah, the last Y chromosomal ancestor of us all. This is basically all a reflection of DNA differences, a reflection of mutations over time. This reflects far too few mutations if deep time evolutionary history were true. We are not looking at tens of thousands of mutations here. We are looking at just a few hundred mutations, as would be expected if the Bible's account of independent origins were true. The data, this data here, did not have to be true, and yet it is. Evolutionists are constantly having to retrofit the data into their evolutionary story. They did not predict low genetic diversity in humans today. They did not predict the one Y chromosomal line, nor did they predict the one mitochondrial DNA line. One last thing to consider is the fact that the length of some of these branches indicate that some people can pick up more mutations in the same amount of time. It's difficult to look at the branch lengths and conclude a definitive amount of time has passed. This is why the pattern and the overall data is so important. It turns out this is what we see for the mitochondrial DNA as well. This is a mitochondrial DNA phylogenetic tree. This is only a reflection of a few hundred generations. Remember, God tells us that everything was created very good. When we look at the modern scientific data, we see a reflection of this. Everything must have started very good right from the beginning. The incredibly complex information storage system of life could not have been built up from inadequacy to precision. If we did not have a highly functioning decoding system, the entire system would come crashing down. Origin of life research has been a massive failure and faces numerous unsolvable chicken and egg problems. Also, evolutionists have no way to introduce functional information into the first life. How do you introduce functional information into random chemicals? If you manage to get the necessary building blocks for life, how is information introduced? This is a major problem for abiogenesis and naturalists such as Jackson here. If I were to spill ink on a paper, I would not at the same time introduce information. A book is more than just ink on paper. A book contains specified information. Also, these next few slides are going to address some of the arguments that Jackson brought up in his opening statement, such as endogenous retroviruses. We just touched on his arguments pertaining to molecular clocks. Let's deal with this data, because this brings me to the fact that we now know the entire junk DNA paradigm has been overturned. I want to point out that the design diversity model, the model that suggests Adam and Eve would have been front-loaded with DNA diversity, which would also apply to the created kinds, would predict that most of the genome and most of these DNA elements found within the genome, including pseudogenes, endogenous retroviruses, ALU sequences, and many other types of variation-inducing genetic elements would be highly functional and active, and this is exactly what we find. And once again, paper after paper after paper. How does Jackson deal with this data? Even the chromosome 2 fusion has been completely overturned. Universal ancestry requires lots of junk and evolutionary leftovers, and we just do not see this. We also have overwhelming evidence for forward thinking in genetics. For example, we have our genes, right, which make physical parts of our cells. Well, we also have what is called epigenetics, which is what controls how the genes make the physical parts of our cells. Our epigenome turns genes on and off, as compared to changing the actual genetic code itself. Our genes are not all turned on at the same time. This would not be good. These are non-random changes. Good designs means we have some type of control, a type of control that makes sure we have the right things being turned on and the right things being turned off. This is all forward thinking, which points us back to the forward thinker. Okay, I want to ask Jackson a few questions here for his rebuttal. And we could also discuss it in the discussion portion. So we know that ERVs, which he brought up, and other classes of retrotransposons accomplish many crucial functions in regulating gene expression, differentiation, development, and even cell stress responses. Does Jackson have any empirical evidence, maybe a technical paper, that actually demonstrates a non-functional DNA element such as an ERV, pseudogene, ALU sequence, going from non-functional to something critically functional in the genome? Praise, how much time do I have, brother? Praise, you are muted. Praise, how much time?
2 minutes. 2 minutes.
Okay. Here's another question I have for Jackson. Human chromosomes are made up of relatively large linkage blocks, and analyses of these original text strings tells us that the genome is young. Why? Because the genome has only been partially scrambled. Crossovers in gene conversion should scramble all linkage blocks quite quickly in an evolutionary time frame. This is a major problem for evolutionary theory. How does Jackson explain this data? A near fully functional genome turns the nested hierarchies and the similarities that Jackson pointed to in his opening statement to evidence for design hierarchies as compared to descent with modification. Therefore, evolutionists have to fight the evidence for genome function, including the results of ENCODE, which suggests that over 80% of our genome is indeed active and functional. If most of the genome really is junk, and these ERVs are nothing but junk, why would the cell bother with so many and so much evolutionary leftovers? Such an activity would be wasteful of energy and resources and would have been eliminated by natural selection long ago. Also, since Jackson liked to bring up the molecular clocks, how does he explain the mitochondrial DNA and the Y chromosome data that, based on the empirical method, tells us we came not from just a small founding population, but a population of just two, Adam and Eve, 6,000 years ago? These are all questions that must be answered for Jackson to have any chance at making a case for human evolution. Why? Because these are just some of the lines of evidence that can differentiate between the models, human evolution and independent origins. That concludes my opening statement.
Perfect. You got right under the time there, SFT. Awesome, man. So now we're going to move into the 10-minute rebuttal session, and I'm going to give you a minute warning beforehand to wrap it up, and then you can have that time to wrap it up. So whenever you begin, I'll begin the timer on whenever you want to start your beginning. Go ahead.
All right, you can go ahead. You mentioned genetic entropy. It's more of a creationist term. I think the real term is error catastrophe, which is when too many mutations accumulate. They've done this in the lab, but in some instances, accelerated mutation rates don't necessarily mean the death of the population. Sometimes it actually includes improvements. I was reading one paper today. I don't have a link to it. But you also mentioned Homo floresiensis and its degeneration. You mentioned insular dwarfism. Well, that's not necessarily a negative mutation, or this is something that happens to a population confined to an island. They tend to get smaller. Sometimes they get larger. In the case of some reptiles, but with mammals, it's usually the other way. They get smaller. Now, for the Y chromosome and the mitochondrial DNA being the same time apart, I couldn't find the original paper, but I found some pieces of it that said that's basically a misunderstanding of some of the original data. Now, low genetic diversity in humans. Some populations have low genetic diversity, but for example, the African population has incredibly high genetic diversity. Abiogenesis, it could be disproven tomorrow, and evolution could still be true. personally, I think life came about through natural processes. I don't know how, and no one really does. They have some hypotheses, but there's no one that's latched onto. Now, ERV function, they don't necessarily have to have a function, or they could have a function, and they can still be leftovers of viruses that indicate common descent. And you mentioned that chromosome 2 was overturned. That's not entirely true. The DNA sequences on the two chimpanzee genomes, of two chimpanzee chromosomes, and human chromosome 2 are essentially identical. And there's a kind of a degenerated centromere. Now, you said, let's see, non-functioning ERVs. ERVs aren't necessarily junk. And the MT and Y chromosome, they honestly don't link up to 6,000 years ago on two individuals. As I've said in my opening statement, from everything I've read, and I couldn't find the paper that said what you're saying. It says over 100,000 years for mitochondrial Eve, and tens of thousands of years later for Y chromosome Adam. Now that's probably a lot less than 10 minutes, but those are the points you touched on that I wrote down. If you have anything else for me to address, I don't know if that's the format or not, but I can. But yeah, that's pretty much it.
Awesome. Thank you for your rebuttal. You have about 7 minutes left over. You can put that in the question and answer period if you want, or your final concluding remark. So SFT, your rebuttal is next. Whenever you want us, I'll start the timer whenever you get going.
Awesome. Thank you. Just 1 second. I will share screen in, probably about a minute or two. So just 1 second. Praise. And I'm just writing things down like crazy, Jackson. Okay. Why don't we start my timer here, Praise. So Jackson talked about afarensis quite a bit. This would be Lucy. He talked about some of the bones that were found. I will touch on this as well. He also talked about Africanus, another Australopithecine. He mentioned Australopithecus sediba, habilis. He mentioned we are related to the chimpanzee and we share a common ancestor with them. He discussed erectus as well. So lots of fun topics here to discuss. He discussed Neanderthals and the Neanderthal genome, which is one of my favorite topics. Humans and Neanderthals are the same species based on the biological definition of species, okay? Since we interbred with them. We literally have their genetics. He also brought up molecular clocks, endogenous retroviruses, and genetic similarities with the great apes. I touched on a lot of that in my opening statement, and I will touch on that a little bit more here. But I want to debunk his fossils first, because Lucy herself was found in hundreds of pieces over a large area. And when they say they have hundreds of specimens of Lucy's species, what they actually mean is 400 pieces of bones. Many of these bones even turn out to be teeth. What we find in terms of paleoanthropology is deliberate exaggeration a lot of the times, okay? Because even Lee Berger, a famous paleoanthropologist, has pointed out that there are more people working in the field than there are actual objects to study. He points out most of the record pertaining to human evolution is bone fragments, bits and pieces of post-cranial bones and teeth, lots of teeth. You'll often, actually, I hear a bit of feedback, Jackson, if you could mute yourself real quick, my good man. You'll oftentimes see these skulls, okay, propped up as, say, Lucy's skull, okay, Australopithecus afarensis, when in fact, most of what you have are a few skull fragments, with most of the reconstructed skull being inference-based. He discussed similarities in bones, similarities in blood types, anatomy, physiology, morphology. Well, as we know, similarity does not equate ancestry, as similarity can also mean designer. It is the differences we need to focus on. And the differences cannot be accounted for by the evolutionists. For example, we've got massive differences in gene content and architecture when it comes to the chimpanzee Y chromosome versus the human Y chromosome. Okay, we also have differences when it comes to epigenetics, genomic networks, and gene expression in the same sequences that the evolutionists say are similar between humans and chimpanzees, and yet massive differences even in those similar genetic sequences. Okay, what about orphan genes, these taxonomically restricted genes that have no evidence for any type of ancestry? They pop up, and evolutionists have no real empirical evidence for these taxonomically and essential genes. How does Jackson deal with these lines of evidence, for example? Okay, we can't just look at the similarities, we got to look at the differences. Now, when it comes to the genetic similarities anyway, scientists, what they did, and you can read about this in the original chimpanzee paper, they ignored huge gaps, size differences, copy number variations. And when we consider both genomes, including size differences and overall architecture, we are looking at something like 80% similar between chimpanzee and humans. Okay, this is hundreds of millions of DNA differences that Jackson here needs to be able to explain as to how they can become fixed. Fixed just means get stuck in place. How can Jackson explain? We got this huge waiting time problem about fixing hundreds of millions of DNA differences in such a small amount of time. Okay, I'd like to point out that if we had the exact same type of blood as Jackson indicated, we should be able to get a blood transfusion with an ape, and yet we know this would result in death. So Lucy, okay, afarensis has been proven to be nothing but an extinct form of ape. She resembles many modern day forms of chimpanzees in arm length, height, skull shape, mandible size, structure, among many other details as well. She, her skull, inner ears, locking wrists, curved fingers, and short legs demonstrate that Lucy was definitely an ape. And as a matter of fact, when you look at the fossil record in general, we see an abrupt appearance. It doesn't help Jackson. We see an abrupt appearance with unique differences in humans. Okay, Erectus has very human-like qualities and unique differences, guess what? Not seen in prior hominins. And this is from its first appearance. Humans have always been humans in the fossil record with no gradual transition from more ape-like creatures to humans. He also discussed Homo habilis, okay? Well, Ian Tattersall calls habilis a wastebasket taxon, little more than a convenient recipient for a motley assortment of hominin fossils. And he believes more than one hominid species is represented in habilis. And this is because Homo habilis is extremely fragmentary and low quality. The existence of artificial species is a real problem for the fossil record. He brought up Sediba. Sediba is the same thing. What we see are artificial species, chimeric species. Habilis is made up of bone fragments, partial skulls, and low quality, low confidence evidence. The defining specimen, for example, of habilis is nothing but a bunch of pathetic fragments. The stone tools that are often found with these bones obviously belong to humans, for one. They have also discovered a human-made shelter alongside habilis bones, okay? Humans coexisted with ape types. We see significant overlap, and oftentimes their bones are intermingled. Considering what was found outside the stone hut when you look into habilis and inside the stone hut, it is clear that what we're looking at is the fact that humans were living in this area, and so humans made the tools. Nomadic people today actually build the same types of stone huts that were discovered around habilis. And the evidence suggests Homo erectus and the Australopithecines lived at the same time. We see significant overlap, as I indicated. We can find evidence that what we are looking at is tribes of humans setting up camps where they made specialized tools and also butchered and ate animals, some of those animals being apes. Humans have always done this, especially in survival situations. And the evidence pertaining to Floresiensis, Neanderthalensis, Erectus, Luzonensis, Naledi, okay, I can go on and on. Floresiensis demonstrates accelerated genetic degeneration due to founder effects and inbreeding, exactly what we would expect post-Babel. Praise, if you want to pause my timer because I want to share screen real quick. So pause my timer, praise. And I'm going to, how much time do I have, praise?
You got 2.5 minutes.
Okay. So what we see here are massive differences when it comes to for one, we've discussed the genetics, but we also have massive differences when it comes to the physiology, anatomy, okay, the bone structure when it comes to humans and chimpanzees. For example, we as humans grow extremely long hair just on the top of our heads, and primates cannot. We have melanin in our skin while apes do not. Okay. We have a subcutaneous layer of fat under our skin, and no ape does. Humans have the same number of vertebra and ribs, and only humans have a forehead, nose bone, and parabolic jaw with toes that align. There are many features that separate us from apes. Okay. In afarensis, here's here's Ardipithecus ramidus. We see that Ardi consisted of scattered fragmentary bone pieces. Okay, a lot of these are due to reconstructions, digital reconstructions. Ardi, when you consider all the evidence, looks exactly very close to a bonobo. And you can see quote after quote from these paleoanthropologists that point out the fact that human evolution is confusing, and all new discoveries make things more confusing. It's a mess. And all of your so-called icons, okay, when you consider their best so-called transitionals, you can see here Ardi, Lucy, Sediba, Habilis, all very, very incomplete, highly fragmented, okay? But when we actually have the data, like we do with Neanderthalensis, Erectus, okay, we can clearly see that what we are looking at is humans, okay? It is safe to consider all the lines of evidence that Jackson point to. What we're looking at, we can easily categorize it in what? Either fully human, degenerated human, fully ape, degenerated ape. Okay, we have Homo erectus here who had evidence for being a sailor, purposeful navigation, was able to speak. This speaks of intelligence. Even today, we see a ton of variation in humans. Okay, you can see the story changing over and over again as the years go on, and the more data that they find. For example, in this chart, you see a bush. It's a bushy mess with no clear transition from ape-like creature to man. Okay, and as you can see, it's low quality. I mean, with Lucy, afarensis, they find this, and they make this. Okay, you've got Lucy with a kid.
You can wrap it up so we can get into our second round of rebuttals.
How much time do I have, Praise?
You can wrap it up.
Okay, so this is the last thing I'll say, and the next rebuttal I'll cover some more stuff. As you can see here, the fingers of Lucy's kind were curved for tree life, and they also discovered a vertebra that belonged to an extinct baboon, and it took them over 40 years to discover this. This is why the fossil record is extremely low quality and low confidence science. Genetics is key to answering this question of ancestry. I yield.
Awesome. So that concludes our first round of rebuttals. Now we're going to go into the second round. It's 5 minutes. It's going to be a little quicker. So if you're prepared to have a little bit quicker rebuttal, that'll be awesome. So whenever you're ready, Jackson, I'm going to start the timer for your 5-minute rebuttal.
All right, I'll go ahead. You mentioned Neanderthals by definition should be the same species because they interbreed, but that's not necessarily true. That is usually the definition of a species, but there are instances of fertile hybrids from 2 different species. Now, from Lucy and her species Afarensis, I did say that most of the fossils are known from fragments, just even individual bones. But there are at least 3 that I know of that are semi-complete that show a good amount of detail between the 3 of them. Now, as for how hard it is to find fossils of these hominids, it took one site, it took them 10 years to find one piece of jawbone. 10 years. So it's not easy to put this stuff together. And you mentioned that the Lucy's skull is fragmentary, which it is, but they've found a male Afarensis skull, which is mostly complete, that shows great detail of what the skull looks like. Now, how do I explain orphan genes? I think was the question. Well, it's not really a problem. De novo genes can arise and be explained just as between generations and getting passed on. And you said that the chimp genome is 80%, which I alluded to that it's not really 99%, but the regions that code for proteins are 99%, so it's not really dishonest to say that we're 99% similar. And Australopithecine arms, you're right, they had curved fingers, they had locking wrists, they had robust shoulders, but their skulls were very ape-like, but pretty much from the neck down, they were very human-like, and I think that's the perfect definition of a transition, to have traits of both lineages. And what came before Lucy, her species Africanus, was Anamensis, which isn't as well known, but and you mentioned that there are some artificial species, like they're not sure about habilis, or habilis rather. They think Rudolfensis may be the same as habilis. They're, again, not 100% sure. Most scientists think they're two separate species. And you're right, it is confusing to make sense of these species. The more they find, the more confusing it seems to get. Zediva and Naledi are good examples. But overlap doesn't necessarily mean they aren't ancestors. Like two different populations of the same species, one can progress and live on, one can kind of go another way and exist at the same time. That makes sense. Now, you showed a picture of Lucy as a knuckle walker. Her hips, her femurs, her knees, her feet, everything about it. Well, she didn't have feet, but her species feet. Everything about it says that it walked upright. It wasn't a knuckle walker. Now, the erectus brains, you mentioned they were small, which was true in the beginning for when they lived in Africa and briefly ventured into Europe, or when they first ventured into Europe. But as they progressed eastward, their brains developed into 1,300 cc's, which is very close to the modern size. Now, Zediva, you said, was very fragmentary, but it's known from two pretty well complete skeletons, and Prometheus is known from a very complete skeleton, exceptionally complete. Let me see. And there are differences in apes, and that would be expected. We're not going to be exactly like them. But the toes of the Australopithecines, or most of them anyway, seem to have aligned with the big toes seem to have aligned with the smaller toes. And that's just more evidence of a transition, I think. An ape-like creature having human-like feet. But I'll agree with you, it is a very confusing lineage and hard to make sense of. It's not really the best transitional fossil line that I would choose, but it is evidence nonetheless. So with that, I yield back.
Praise.
Oops, I'm sorry. Yeah, on mute. But yeah, perfect. You were 15 seconds to spare, Jackson. Great job.
Right.
So now we'll hand it over to SFT for his 5-minute rebuttal, second round. And then we're going to go into 25-minute open discussion. Yeah. So whenever you're ready, SFT, I'll start the timer.
All right. Yeah, you can start it now. So many things to address. I could probably go on for 10 hours. I'll start here because Jackson's spending a lot of time on Lucy. Okay, afarensis. Well, even admitted by Lee Berger. Okay, Lee Berger has admitted that Australopithecus afarensis is a chimeric species. It is a jumble of bones from multiple species. Even Donald Johanson has reported at one point that at least 2 different species from 2 different genera were represented in his bones. Some fossils even looking exactly like human fossils. I'll tell you what happened, okay? And this is a fact. Due to the extensive range of differences seen in the bones attributed to the Australopithecines, including Afarensis and Africanus, what they invoked was called sexual dimorphism. This is simply the physical differences in males and females of the same species, okay? So paleoanthropologists claim that the larger human-looking bones belong to larger male Australopithecines, while the smaller ape-looking bones belong to females that lived in the trees. And this is why Jackson can focus on those bones, which, by the way, we don't have any genetics. Fossils and bones are not inherited sperm and egg. Geography is not inherited sperm and egg. It's the genetics, the genetic data that confirms independent origins. For example, when it comes to genetic degeneration, when it comes to Y chromosomal data, mitochondrial DNA data, but we'll get more into that a little bit later, okay? So people like Jackson, they can look to the larger bones or the bones that are actually human, since Lee Berger himself admits that Afarensis is a chimeric species, and point to those bones and say, see, Lucy had some human-like qualities, when in fact what they're looking at are human bones. Okay? Everything in human evolution is contested, and he agreed with this. And even this whole sexual dimorphism that is invoked is not a 100% agreed upon claim. Now, many proponents of human evolution have conveyed disagreement with pretty much everything in terms of human origins, okay? And the original assessment that Afarensis was a jumble of mixed bones, including human and ape bones, is clearly more accurate, okay? To put it simple, arboreals like to emphasize the bones that have been called Lucy's kind that look very apish, while others will emphasize more of the isolated bones, because remember, the fossil record is extremely fragmentary, and you find these isolated bones that look human and say this isolated bone is more human-like, and therefore Lucy walked upright like modern humans, when in fact this is all better explained by the chimeric nature of Afarensis, these artificial species. Christopher Root, for example, co-author of Contested Bones, points out that we have never seen a case of sexual dimorphism where the male locomotes so much differently than the females. This is the nature of chimeric species and the mixing of these bones. I'm not saying it's done deliberate, okay? This is oftentimes accidental. Now, I want to share screen real quick and go over a couple more slides. If you can just stop my timer real quick. And right here. Okay, so when it comes to Sediba, okay, we have many paleoanthropologists. Here's a paper here. We have paleoanthropologists that have pointed out the fact that Sediba may be an artificial species, meaning the accidental mixture of human and non-human bones. Sediba and habilis being artificial constructs because they consist of a loose collection of human and ape bones. Ella Bin, for example, and Yoel Rack hold this view based on the lumbar vertebra and jaw bones that are derived from two different genera. Okay, and yes, you do often find Sediba with some articulated bones, but this is the point, because Sediba has been described as anatomical contradictory. For example, upper limb and shoulder anatomy are just like arboreal dwelling orangutans, and then they've got hands indistinguishable from modern humans. You see a paper here, human missing link fossils may be jumble of species. So this is what we see. For example, we also have evidence for human footprints. Okay, we got the Laetoli footprints. Well, years ago, Mary Leakey discovered footprints that are indistinguishable from modern human footprints. As a matter of fact, if these footprints were discovered today, nobody would question these footprints. They would all concede. They would all conclude they were made by barefooted human beings. Evolutionists have dated these footprints to roughly 3 or 4 million years old, and therefore, because of their circular reasoning, they cannot conclude these were made by humans. What's that, Praise?
Go ahead and wrap it up.
Okay. We also see the same thing in Crete recently, anatomically modern looking human footprints. And last thing I'll say, a recent finding here that 3 different early humans, Australopithecus, Paranthropus, and the earliest known Homo erectus, appear to have lived at the same time in the same place. The point is, overlap is what we would expect from a biblical model. And that's exactly what we find. I yield.
Great job. So now we're going to go into the 25-minute open discussion. You guys can just bounce off that if you wanted to, and just SFT's last point, Jackson. It's up to you guys how you want to do that. But whenever you guys, I'll start the timer.
All right. So you say Afarensis is a chimeric species, but I think that doesn't really work, given that there are three that I know of, three partial skeletons found at the same different sites, and all together, mostly together. Lucy was found in a 50 square foot area, I believe it was. I mean, it may be meters, but something like that. But the other two were found much closer together. That's from what I've read. So,
right. So,
what do you think of that?
Yeah. So good points. You would agree that Lucy herself was found in hundreds of pieces, though, over this area, right? Very fragmented.
Mm-hmm.
And Lee Berger, I'm not sure if you're familiar with this, but even Lee Berger has pointed out that Australopithecus afarensis may indeed be a chimeric species, as in it is a jumble of bones from multiple species. And Donald Johanson, at one point, has made the claim that we are looking at least two different species from two different genera. Okay, and he was pointing this out in the bone collection that he had, with some of those fossils even looking exactly like human fossils. For example, I'll point to I'll share screen real quick. The Laetoli footprints, which I'd be curious as to your thoughts, where we've got footprints here in Laetoli that are indistinguishable from human footprints. So what is your position on this? And why not conclude that, since, as you can see here, here's a paper by Donald Johanson verifying what I was saying here, this collection suggests that Homo and Australopithecus coexisted as early as 3 million years ago. So why is this not a human footprint, since that's exactly what it looks like? Yeah, what are your thoughts on that, Jackson?
Well, I think radiometric dating kind of shows that it couldn't be a human. I know you guys don't really give a lot of credence to radiometric dating, but also the foot structure of what we know to be afarensis, not from Lucy, obviously, but the fourth metatarsal, the heel, shows that it probably didn't have a divergent big toe, and it had an arch and a striking heel for bipedal walking. So I think the Laetoli footprints may have been afarensis. It may have been another species, but I think they weren't human.
But do you find that circular, in that we have paleoanthropologists that are pointing out, for example, here's the footprint, here's a human footprint. We see how obviously divergent it is from a chimpanzee footprint. We don't really know what Lucy's feet look like, because what we have found is very fragmentary. So therefore, it seems to me that this argument is circular, Jackson, because you pointed to dating methods, which I'll go to, which we know are based on many assumptions, many of them unprovable assumptions. And I've got a slide here that goes over numerous sources from rocks of known age, as in we know when the volcano erupted, the samples were collected, and we get dates of millions and millions of years old, even though these rocks were just formed. So the point is, why would we trust dates for rocks of known age, right, or unknown age, when we have rocks of known age that are giving us inaccurate dates? I know this isn't a debate about dating methods, but my point is, your reasoning seems to be circular, because you're pointing to, well, it's dated far too old to actually be humans, instead of concluding the obvious. Yeah, go ahead.
Well, I know that the ICR did a test, the rate test, the rate study, and they used potassium argon dating to date Mount St. Helens, which was the incorrect method for that. It even said that Geochron Labs they use to date the sediment says anything below 2 million years, it's going to give you wild answers. And they did get multiple different answer ages back. And they think that's probably from, what's it called, ion noise, I think it's called, from the mass spectrometer. So I think you can dismiss the ICR's research on that. And I know there are instances of multiple dating methods aligning on one sample and coming to within a few percent of the same age, which is very unlikely if it didn't actually work.
So what's nice about the rate project and the rate scientists is they took samples from the same rock layers and subjected them to the dating methods all at once. And what they found were many discordant dates, even with the isochron method. And these were repeatable. Okay. And they also did this with C14 carbon dating. And what they found was carbon 14 in diamonds, in fossilized wood, in dinosaur fossils, in coal, which means these samples cannot be millions of years old or billions of years old in terms of diamonds, because obviously we're finding C14 in these samples when given the half-life of carbon 14 and the fact that carbon decays so quickly, then obviously these rocks and fossils, okay, that the uniformitarians and evolutionists are pointing to as millions of years old, that kind of goes back to your argument here, which I believe is circular based. These samples clearly cannot be millions to hundreds of millions of years old because they had radiocarbon in them. And you can comment on that, but I did want to ask you this question. I'll get this question out first because I kind of want to bring us back to more of the topic, but I also don't want to leave that hanging. So you can definitely address that. But I wanted to ask you the question in terms of genetics and the fact that we have massive differences, which I discussed in the opening statement and my rebuttal, but a huge difference has to do with the Y chromosome, where we've got, I'm not sure if you're familiar with this paper, chimpanzee and human Y chromosomes are remarkably divergent in structure and gene content. When you consider overall size differences and architecture, Jackson, we're looking at a Y chromosome that is less than 35% dissimilar between humans and chimpanzees. So if they are our closest cousin and we share a common ancestor with the chimpanzee, how do you explain those huge differences? And take your time, take equal time, Jackson, go ahead.
Well, the differences in the Y chromosome, I know the Y chromosome mutates a bit faster than the other chromosomes, I believe. And as for carbon 14, again, it was likely due to contamination because uranium and thorium decay and can produce carbon 14 in the surroundings. So the fact that it was there doesn't necessarily mean it was ancient. So that's pretty much my comment on that.
Yeah, so what I would say is the rate scientists, what's good is they went into the rate project, okay? They went into it making predictions. And what they discovered were fulfillment of those predictions. For example, they even found helium and zircon crystals. We know helium is incredibly slippery, and yet we find helium and zircon crystals that evolutionists say are billions of years old. There should be no helium left. Same with the radiocarbon. So you point to contamination, but what's good is the rate scientists were diligent, and they subjected these samples to harsh treatments that would get rid of any of these contaminants, and therefore what they're looking at are blank samples. Okay, so they've looked at contamination, they've looked at other reasons that the evolutionists might point to in a way to rescue uniformitarianism in deep time, and these results are repeated. These are repeatable. They're testable. We're finding radiocarbon oftentimes 100 times above the limit the detectable limit that should be there. When it comes to the Y chromosome, the problem is in, let's say, 6 million years, you've got such a waiting time problem to account for massive differences in the Y chromosome. The Y chromosome is uniparentally non-recombining DNA, okay? It is inherited from our fathers. The problem is it should be relatively more stable due to the fact that it's non-recombining. But as you pointed out, the Y chromosome also mutates fast. So here's a problem. If you are going to look to that argument to explain the dissimilarity between human and chimpanzee Y chromosome, well, we have a lot of papers telling us now that since the Y chromosome mutates faster than ever predicted, there are only a few mutations in the Y chromosome separating any two people on the planet. When it comes to men, every single one of our Y chromosomes are nearly identical. So how do you have a rapidly changing Y chromosome with incredibly low variation in humans today? Every single human, every single male Y chromosome is nearly identical, and they can be traced back to one Y chromosomal ancestor just 4,500 years ago, as you can see here. So how would you address that if you're going to look to the rapidly changing Y chromosome? Why do we see very low Y chromosomal variation in the male Y chromosome, Jackson?
Well, I would say it's because humans are a relatively young species, no more than 200,000 or 300,000 years old. So not really much time relative to every other animal to acquire the sufficient mutations to look as different as you would expect, I would think.
Well, here's the person. Oh yeah. I'm sorry. Go ahead, Jack.
As for the helium and the zircons, they tested a sample that was taken out in the 70s, I believe, and it was taken out from a volcanic area. And from what I've read, the compression of the zircon can hold in helium for long periods of time, and the system isn't well understood in the first place to really assess the age from that, based on what I've read. And now some form of helium will decay past that point, but some forms of helium, like helium-3, are stable over long periods of time. Now, what was your next point?
So my point would be, and we can touch on that in a second, okay?
Okay.
My point here would be, here's a Y chromosomal family tree, okay? And this is based on worldwide Y chromosomal diversity. We're not looking at hundreds of thousands of years here, okay? We're not looking at tens of thousands of mutations to take your Y chromosomal Adam ancestor into the deep time that you need it. No, all we're seeing is a few mutations in the Y chromosome that separates any two people. Okay, let me ask you this question, okay, just to kind of steer this into relatively something new, okay? We don't only have low variation in the Y chromosome and the mitochondrial DNA. We have low genetic diversity in general, okay? Let me ask you, Jackson, what happens when you introduce a new mutation into the genome? What does that add?
Usually it has no effect. Most mutations are neutral. Some are harmful and some are beneficial.
Well, we'll get into we will get into that, because I've got a ton of quotes where population geneticists that are evolutionists admit that no mutation can truly be neutral. Okay, they refer to these as effectively neutral because they have only very subtle effects on fitness. They're only slightly deleterious, kind of like rust on a car, okay, that they build up over time, but they're still unselectable, and therefore they're subject to genetic drift. So they accumulate and degenerate. But that's separate, that we can get into in a bit. But just in general, mutations. When you introduce mutations, what do they add?
Well, it depends on the mutation,
according to the evolutionary model. What's that?
I guess it would depend on the mutation. Like there's a de novo mutation in cod that allowed for them to live in colder water, for their blood not to freeze.
Well, how do evolutionists explain the origin of genetic variation and genetic diversity?
Well, what do you mean? Like from the
Like,
beginning
what are
of
the
time?
mechanisms? Like for example, we
Mechanisms?
as creationists, we look to what's called the created heterozygosity hypothesis, the design
Hmm.
diversity hypothesis, that suggests that the vast majority of nuclear DNA differences are the result of created heterozygosity. Heterozygosity just means a state of DNA diversity, while the evolutionary community explains the vast majority of DNA differences as mutations, right?
Hmm.
Okay.
Yeah.
So when you have a mutation, according to the evolutionary model, what are you adding?
Well, oftentimes new information. I mean, there are mutations that de novo mutations, duplication mutations that add material or new information to the genome.
So, in other words, when you add a mutation, you're adding genetic diversity to the
Well,
population.
not necessarily diversity, but information. Yes.
Well, the evolutionists explain the vast majority of DNA differences, if not all DNA differences, and the origin of genetic diversity as the result of mutations over time. Okay, so a mutation adds something because it is adding something that was not there before. So I'm just going to go right to my point then. Okay.
Okay.
If you have the Australopithecines evolving for millions of years and you got Homo habilis, okay, and then you've got what I find interesting is I've got a couple articles that I can show you later where
Okay.
Australopithecus sediba has been ousted out of the human line due to some
Yeah.
contradictions that they discovered in 2019. So let's just throw away sediba for now. We've got
Yeah.
habilis, and
Sadiq
then
was a
you've got
weird case.
erectus. And then you got erectus living on the planet, covering part of the globe, Africa, Eurasia, for millions of years, where they are also accumulating mutations. So we should have today in humans high levels of genetic diversity. But what we find is low levels of genetic diversity. Why is that the case?
Well, I think there's higher genetic diversity than you're letting on. But there was a genetic bottleneck in the past, about several thousand individuals, as evidenced by mitochondrial and nuclear DNA. So I think they can explain a lot of the low genetic diversity.
So here's the problem with that. And I'm not going to screen share again. We'll just have a free flowing dialogue. But I do
All right.
have papers in front of me that go over the fact that we do have extremely low genetic variation. As in every single human being, we're all 99.99% similar.
Yeah, that's true.
So that was a problem for the evolutionary community. And you gave the correct answer, according to the story, is in the past, there was a genetic bottleneck that reduced the population of at the time to roughly between 2 and 10,000. How many years ago would you say this was, according to your model?
I believe it was about 80,000 years ago.
Okay, so say you've got.
That was the case.
Okay, so say you've got, because here's the thing, there's differing opinions as to when this bottleneck took place. Some people say, a couple 100,000 years ago, maybe 70,000 years ago with the Toba extinction, or some even say millions of years ago with Erectus. But let's go with 80,000, okay? So
All right.
here's the problem with that is you've got this bottleneck that drops the human population to between, let's say, 2 and 10,000 years. Well, you would need thousands of years. You would need more than one generation to reduce those levels of heterozygosity and increase levels of homozygosity, okay? Therefore, you have generations and generations of what, Jackson? Of inbreeding. And is inbreeding a good thing?
Well, no, but by your model, inbreeding is the only option also. I mean, inbreeding from a point of a few thousand is better than inbreeding from a point of 2.
But here's a question I'm going to ask you. In light of what we've been talking about, what is the major difference in terms of the origin of genetic diversity between the evolution model and the creation model? What's the difference?
Well, I think the creation model says all the genetic diversity was put in at once, in Adam and Eve, and the evolution model would say new genes can arise.
So the creationists would say that the origin of genetic variation for the most part is due to design diversity, while the evolutionists would look to mutations as the source for genetic variation. So here's the problem. We don't explain every single DNA difference as being the result of mutations over time. So Adam and Eve would have had pre-existing functional heterozygosity with no mutations. And then at the flood, you might have some mutations built up, but still what you're looking at is mostly created variation, okay? It's the mutations that are in recessive form that are manifested during inbreeding that lead to accelerated genetic degeneration. It is the evolutionist that proposes mutations as the source for genetic variation and genetic diversity. So you've got this bottleneck roughly 80,000 years ago where you've got all of these mutations, these deleterious mutations that are now coming to the forefront, leading to rapid genetic degeneration. Because it's not one generation, it's not two generations, it's many generations of this inbreeding in order to reduce levels of genetic diversity. Are you familiar with the bottleneck today that is seen in cheetahs?
Yeah, cheetahs and Tasmanian devils and a few others.
So we see in cheetahs how disastrous this is. There's about 7,000 cheetahs today. Birth defects are increasing, and the cheetahs are essentially going extinct. Conservationists are extremely worried about them. So what we see with the cheetahs means that this hypothetical out of Africa bottleneck would have also been disastrous. So my question to you is, how is that bottleneck that's employed to explain the low levels of genetic diversity, which, by the way, is what we would expect starting from Adam and Eve, that automatically would restrict diversity. How is that hypothetical bottleneck associated with massive problems with inbreeding? How is that feasible, would you say?
Well, biologists say, or zoologists say, that the minimum population for optimum genetic diversity is about 100 individuals. So we would have had a few thousand to work with, so not catastrophic. But I also want to say, what you said, Adam and Eve wouldn't have been subjected to mutations. Is that right?
At creation, they wouldn't have had mutations, right?
Yeah, but after the fall, would they have?
Well, the fall would have introduced mutations because mutations are essentially copying errors.
Yeah.
So due to mutation accumulation, we inherit roughly 100 new mutations per person per individual. Over time, Adam and Eve's descendants would have had mutations. Yes.
Yeah, I think that's a way less workable model, just because you're starting from 2 and you're just inbreeding immediately from their children. And any gene is going to get expressed and passed on, and it's just going to be kind of a total mess.
Well, let me tell you why. So it wouldn't be a problem from our model, because according to the independent origins model, inbreeding would have been a non-issue since there would have been no deleterious mutations. And since mutations can be compared to typographical errors in a text, and during instances of inbreeding, those mutations come to the forefront, leading to disease and damage. But if there are no mutations to come to the forefront to lead to that type of disease and damage, then we wouldn't have an issue. But that brings me to the point you made on neutral mutations. And you said that a mutation can not be deleterious. I'm trying to find, I got 1,000 slides here, even more, 1,000,000 slides. So here's a source, okay, from, because you said it was a young earth creationist invention or talking point. But we have population geneticists here that point out, for example, right here, Higgins and Lynch, we find that the accumulation of new mildly deleterious mutations, okay, so mildly deleterious, and this one here, Crow's concern, the typical mutation is very mild. It usually has no overt effect, but shows up as a small decrease in viability and fertility. And the last one I'll show you is right here with Kondrashov. He points to very slightly deleterious mutations. There's also another quote that I can show you where a population geneticist points out the fact that any mutation must have at least some effect on genotype. So how do you explain if those mutations are only mildly deleterious and they build up, Jackson, which means they're unselectable? What
All
type
right,
of
guys.
selection can remove?
We got to start wrapping it up. We just finished the 25-minute mark for the open discussion. So we're going to go into the speed rounds and then your conclusions. So that was a amicable and enthralling discussion. Good job, guys.
Why don't we?
Yeah, I think it was good.
Yeah, I want to give Jackson the floor here, though, just to answer that. I don't want to end it there. So, Jackson, so the question would be, what type of selection can you put forth that would remove so many low impact deleterious mutations? And then take your time responding.
Many of the neutral or slightly harmful, as you would call it, mutations would build up. But the harmful ones, the actual harmful ones would be selected out by natural selection. And I want to bring up the tuatera, not quite a lizard, but it is lizard-like from New Zealand. It currently has the highest mutation rate of any animal, acquiring new information, as well as having some of the neutral mutations. Fastest evolving animal on the planet at the moment. So it just shows that the genome can kind of grow like that and not really have negative consequences. Also, you mentioned cheetahs. I didn't go into that, but cheetahs, it was thought that their population was only down to a few individuals at one point. That's why they're having such problems now. But humans had a few thousand individuals, so much more to work with. Now, starting from Adam and Eve, again, that would have been pretty problematic for genetics. And with that, we can go to the next subject if you want.
Okay, I appreciate that. Praise, are we doing okay? So we're going to do 2-minute responses now, Praise, to wrap up the discussion. And then we're going to go into 5-minute concluding statements. Since we did hit the hour-and-a-half mark, we agreed beforehand. We're going to have a 2-minute intermission right before the 2-minute intervals and then 5-minute intervals, and give you a chance to gather any questions that are coming through. I know we've got a big chat. And here we go, Praise. 2-minute break.
Are we on break?
Yes.
Okay. Can the audience hear us or no?
Yep. So you have to go hit the bathroom. Go do it now.
Okay. I'm good. I'll just sit here.
Okay, it looks like we are good. Praise, we're going into a 2-minute response. Who starts, Jackson or myself?
So it would be you because you opened up, Jackson opened up, so it'll be you in the speed round.
Okay, so I start the 2 minutes?
Yes, I'll start the 2 minutes on your first word.
Okay. Just 1 second. I want to share screen. And okay. So here's the problem. To kind of give some final thoughts on the open discussion is when it comes to the evolutionary model, they explain the origin of genetic variation as being the result of mutations. Since we know mutations are destruction, okay, they lead to disease. And as we can see from many population geneticists, they're acknowledging that most mutations are effectively neutral. Okay, they are unselectable as they build up in populations over time, 100 new mutations per person per generation, and they lead to genetic damage. So that bottleneck that the evolutionists look to as a rescue device to explain why humans today have such low genetic diversity, consistent with the Adam and Eve starting point, is not even remotely feasible, because it would lead to rapid and accelerated genetic degeneration. We see that in the cheetahs. We're not going to suddenly expect these 7,000 highly degenerated cheetahs today to spread out into all parts of the globe, seizing dominion over the planet. Of course not. But the evolutionists want us to believe that after this bottleneck that would have been highly damaging, humans spread out in all parts of the world, seizing dominion over the planet. It just wouldn't happen. They would be highly degenerated. It wouldn't be possible. So it is a fact. Here's a source. Mutations are the source of genetic variation for the evolutionists. But with the icons that Jackson points to, we find this problem of mutation accumulation explaining a lot of the anomalous features that evolutionists like to point to as transitional or anomalous. For example, Homo floresiensis, Neanderthalensis, erectus, naledi. Okay, this is what we find in the fossil record, is where we find inbreeding, we find pathology and disease and reductive evolution. Last thing I'll say, this is consistent with the post-Babel dispersal, and it's inconsistent with human evolution, ape to man evolution. Go ahead.
Go ahead, Jackson. We'll start your 2-minute timer, and I'll give you a 20-second warning on your first word. Go ahead.
Well, I think the genetic bottleneck argument is far worse for the creationist point of view. Not only do you start with Adam and Eve, but then 2,000 years later, you have Noah and his family. 8 individuals, 3 breeding pairs. Or if Noah did Noah have a kid after the flood? I don't know. But anyway, let's say 4 breeding pairs. So it's reduced again. So you're inbreeding yet again after Adam and Eve. And not only that, but all animals on Earth would have been highly inbred. It's not just cheetahs or Tasmanian devils that would have shown evidence of this. Every animal should be showing signs of having reproductive issues. I think I read that 90% of cheetah cubs don't make it to adulthood. That should be happening to all animals if that were the case. Now, so I think the creation model for genetic diversity just doesn't really work, and I think an evolutionary model is definitely more workable. And with that, you can respond.
Actually, you have your conclusion, your concluding remark. It's set for 5 minutes, but you guys did go under your expected time, so you can add a little bit in there. Maybe 5 to. I think it's set for. Let's see here.
Yeah, maybe
5 minutes. You can go 5 to 7 minutes is fine.
Okay, so since Jackson started the debate, Jackson, take the floor. You have between 5 and 7 minutes for your concluding statement.
All right. Well, I think we both did pretty good in this debate. I think it went really well. I know I enjoyed it. We should do it again sometime. But I think that fossils, genetics are really on my side when it comes to this. And I don't know if I need the full 5 to 7 minutes, but yeah, I just really want to say that I enjoyed being on here, and it was fun. And you made some good points standing for truth. You're very competent, you're knowledgeable. So I have a lot of respect for you. Just want to say that. And honestly, that's all I have to say as a closing statement.
Awesome. I appreciate that, Jackson. Okay, Praise, I guess you can start my timer in 1 second here. And I'll share screen in a second because I do want to mention one thing. So you can start my timer now, Praise. Jackson, yes, thanks so much for doing this debate. I
Yeah,
respect
you're
you as
welcome.
well. This was very cordial, and
Yeah.
time has flown by. I can't believe it's already been an hour and a half. It's been fast paced,
Yeah, that's flying
and
by.
there's just so many topics to discuss and so many lines of evidence we could discuss that let's definitely have another one of these in the future. I know we're working on setting you up with debates with Ken Hovind, the film free. So this should be fun. So in terms of my concluding statement, then I'll just address a couple things that I think were left hanging, because when it comes to the fossil record in general, okay, when we look at humans in the fossil record, we see no evidence of hundreds of thousands of years of human existence, okay? And given the evolutionary model, we can assume there were about 1 million. Let's look to Homo erectus, the Australopithecines, Homo habilis, Homo erectus, who lived apparently across Eurasia and Africa, okay? Essentially, they handled a massive distribution of the earth. So if we put some numbers on it, let's say Homo erectus's average generation time is 20 years, let's say, okay? That means about every 20 years, you have another 1 million dead bodies. And so after 1 million years, we have over 20 billion dead people, okay? Point is, maybe it's not quite that high, but we've got a lot of dead people, a lot of dead erectus. And the point is, this is millions to billions of people of which we have no evidence ever existed. Even the proponents of evolution would agree that erectus had primitive technology. They would have had campsites, they would have had villages, they would have had fire. We have a ton of evidence for their existence, and yet every single erectus fossil that we have can be fit in the trunk of a car. So where's the evidence that human evolution even occurred? Millions of years of human evolution. I want to share screen real quick here as well. Let me know when we reach the 5-minute mark, so then I can kind of wrap up from there. Okay, so when we look to because we discussed dating methods. See, here's the thing. Time is always looked to by the evolutionists as the hero of the plot for the story. But what we know about mutation accumulation and genetic degeneration, okay, time is not the hero of the plot, because the more time you have, the more extinction and degeneration you have. So they can have the time. But what we see here is hundreds of physical processes that set limits on the age of the earth. And we discussed just a few, the C14, we discussed helium, we discussed the discordant dates in dating methods, as well as in isochron dating, which they like to look to. We've also seen a lot of discussion pertaining to Lucy, and what we can see here based on her skull, based on the hundreds of pieces that were scattered across this 3-meter area of a hillside, the fact that what we're looking at is highly fragmentary data means you can have all sorts of interpretations and stories with these bones, okay? Bones found in the dirt, fossils, I always point out, they can oftentimes deceive us, okay? Because they don't come with genetics oftentimes. And the problem is, we know that today, many species have more variation within themselves than between species. So when we're looking at a fossil without genetics, without genetic data, then it's oftentimes difficult to conclude, is this the result of homology or convergent evolution? Convergent evolution is the independent origin, apparently, or evolution of highly sophisticated and complex traits that we have no evidence could evolve in the first place. But evolutionists say that something like the eye has evolved dozens and dozens of times. Okay, anytime convergent evolution is invoked, it is being invoked, and it is an admittance of the lack of uniqueness in the phylogenetic tree, okay? So we've got Lucy here, semicircular canals, better suited for walking on all fours. I pointed out the fact that they found a baboon bone, okay? It took 40 years. Homo habilis, evidence that Homo habilis is a chimeric species. And the problem with the taxonomically restricted genes, the orphan genes, they're highly essential, and you would need an extraordinarily amount of mutations that are beneficial, okay, in sequence to evolve something so essential. But yet what we see...
You're at 5 minutes, SFT.
Thank you. I'll start winding it down. What we see with the presence of these genetic sequences that are functional and essential is no apparent relatives, as in it's exactly what we would expect to find according to the independent origins model. And the last thing I want to cover, so I want to make sure I cover everything, is the retrotransposons, which he spent some time talking about. So the question is, when it comes to these retrotransposons, is are they really the ancient remnants of viral infections, or are they created units of DNA function? Because Jackson made the argument in his rebuttal that we still share more ERVs with the chimpanzee and the humans. Humans and chimpanzees share a lot of these viral-like elements than with other species. But that's exactly what we'd expect to find, is if these were created, okay? If these variation-inducing genetic elements are there for a reason, and that's what we find, paper after paper, retroviral promoters in the human genome, then of course we're going to share more, because we predict function, because we predict that these similarities are there for functional purposes. And that's what we see in the design world. Engineers build in homologous patterns. Engineers build in nested hierarchical patterns. Engineers even build transitional-like forms. For example, an amphibious assault vehicle and some of these crossover SUVs. So what we predict is function. What we find is function. Scientists identify new beneficial function of endogenous retroviruses in immune response. The last thing that I'll touch on, I don't want to leave hanging, was the chromosome 2 fusion that he talked about. Well, what we find is that the chromosome 2 fusion, the so-called site, is overlapped by a highly functional and essential gene. So Jackson, the problem is you're not going to get a highly functional gene by slamming together two chromosomes. The site is very degenerate, and there's lack of evidence for the cryptic centromeric site, as well as a satellite DNA problem that maybe we can get into in a future show. This would be a paper I'd highly recommend: The Evolution of African Great Apes, Subtelomeric Heterochromatin and the Fusion of Human Chromosome 2. Chimpanzee and gorilla chromosomes differ from human chromosomes by the presence of large blocks of subterminal heterochromatin thought to be comprised primarily of arrays of tandem satellite sequence. Why are these not found? Why is the evidence seem to suggest there was no fusion event to begin with? And great debate, lots of fun, great chat today, and I hope we can do this again in the future, Jackson. Thanks so much. I conclude my statement.
Yeah.
Awesome, guys. That was a very respectful and stimulating debate. Great job. Both sides made great points, and yeah, it was impressive. But we're going to head into the question and answer period with the super chats. We got quite a few, so we're just going to jump right into them. So the way we format the Q&As and super chats is whoever was addressed to, they get to finish the final comment on that comment, whatever question or comment, whatever it is. So this is from Cheryl for $5. The comment, she says, "This is in the name of Raymond or Ramen Goiza
Ramen, the super sticker master,
who could not be here. We are thinking of you!" Exclamation, exclamation mark. And I actually lost that super chat, so I appreciate Cheryl giving it back. So thank you, Cheryl.
I appreciate that super chat, Cheryl.
So we'll go to a super chat from One Cow Stampede for $10, and thank you for your super chat. And then we'll go to the next super chat from Landon Freeman from $2. This is addressed to you, Jackson. What evidence would change your mind?
About evolution or about God, or what specifically?
Either. Yeah, he didn't specify it, but probably the debate, probably evolution. Yeah.
Evolution, yeah. Well, if you found, say, a bunny in the Precambrian dated rock, that would convince me pretty quick that something's wrong. Or if you could find another animal that had a closer DNA coding than a chimp to a human, like say a pine tree had a closer genetic match to humans than the chimps did, that would change my mind pretty quick also. So it's evidence like that that would totally turn evolution on its head.
Gotcha. So SFT, do you want to say something to add to that, or you want to go to the next? This is the next super chat.
Yeah, can you repeat the question, brother?
What evidence would change your mind? So if it's applied to you, what would change your mind about creation or intelligent design?
Just like I write in my books, there's two predictions, okay? Evolutionists predict, if this is all just a long history of descent with modification and universal common ancestry, then we should find a lot of junk. We should find evolutionary leftovers. We should find genomic fossils. We should find ancient viral infections littering our genome. And yet, what we are now finding is that the vast majority of DNA elements, DNA sequences, and DNA in general is active, functional, and essential. Okay, so what would help to change my mind if that was not what we seen? If we actually did see the vast majority of DNA was junk and therefore reflective of universal common descent, then that would be convincing that banana plants and whales are related. It's just not what we see. And the last thing I'll say is this. This is why evolutionists struggle to make predictions on DNA function, while creationists expect function. So we can look to say the endogenous retroviruses and predict function. We can predict function in determining cell types, in gene regulation, in immune response, cell stress response, and so on and so forth. Same thing with the pseudogenes. And that's it.
So you can finish that, Jackson, if you want to respond to that really quick, a pithy statement if you want to respond to that.
Well, with the ERVs, a lot of them are highly fragmented and evidence of a long time of deletion mutations and inactivating mutations throughout time, and apparently are ancient from everything I've read on it.
But if I could ask you a question, Praise, before we move on, it's just going to be a question. We won't debate it. Have you seen a paper? Have you, for example, because what we're seeing in terms of the amazing functionality of these retrotransposons, are you familiar with any empirical evidence or any technical paper that actually shows a viral-like element, Jackson, going from non-functional to functional? Have we seen this in real time? Is this empirical?
I'm not aware of a paper like that. I haven't really researched that aspect of it. I'll be honest with you on that.
And I think that's the problem, because I've debated a biologist in the past who also looked at ERVs. And there's a number of critics that I've debated where I've constantly sought a paper like that, because it would be incredibly difficult to have a viral-like element, if it was truly negative to begin with, evolve something so essential to the genome. Because if I were to have somebody blindfolded and tell them to randomly put some brush strokes on a painting, well, they're probably going to mess up the painting as compared to improve it. And that's what we see with these viral-like sequences. If these really are insertion events over millions of years, they should corrupt as compared to make things better, especially make things better in the placenta and in embryological development. So until we see an actual paper or technical data that can show, a non-functional retroviral-like element going from non-functional to functional, then my point would be it's just not science. And ERVs shouldn't be looked to anymore by the evolutionists as evidence for common descent. And Jackson, you can have the last word on that. Go ahead.
Well, endogenous retroviruses, I would assume they're like mutations in the sense that most of them aren't going to have any effect. Some of them might have a negative effect, and some of them might have a positive effect. But other than that, I don't have much to say on that aspect of the ERVs.
Very good. So we have Jerome Super Chat, SFT with a Popeye flexing emoticon. Yeah, $5, Jerome. Thank you for your support.
Thank you, Jerome. Thanks for the support, brother. I appreciate it.
Now we got two. So we got a super chat and a super sticker from Jamie. Went for $1.49 and a $5 super chat. He says, he asks, is common ancestry falsifiable, genetic similarity as is? If yes, how? If no, why is that a problem or not?
That's for me?
It's for both of you. Whoever wants to, address that. I think it's addressed to both of you guys.
Well, yeah, common ancestry would be falsifiable. Again, if you found that the genome didn't match close to chimps, it matched, say, to pigs, closer to pigs than chimps, that would turn common ancestry upside down. It would, just really anything like that would disprove it. So it is falsifiable.
For my response, I would just say it's not falsifiable because we have so many lines of evidence that have overturned universal descent, okay, universal common ancestry, especially in the fossil record where we find modern human bones, modern human footprints in areas where they should not be. Because Jackson said earlier, and it's a good point, I agree. For example, we can live at the same time as our parents and grandparents, okay? A domestic dog, although ultimately came from a wolf, can still live with wolves, right? We have wolves and we have the domestic dog living together, okay? The problem is you can't have a domestic dog living before the wolf. We can't have me living before my great-grandparents. And that's oftentimes what we find with this overlap in the fossil record when it comes to modern human-looking footprints, when it comes to the overlap we see with the Australopithecines, with Homo erectus, okay, with all of these so-called hominids. And it just better supports the post-Babel model, where you've got these people groups that migrate to different parts of the world, and we see similar anomalous features in Naledi, in the hobbits, right, Floresiensis, also in Luzonensis, where they experience island dwarfism and reductive evolution. Reductive evolution seems to be the rule, and reductive evolution is not going to take a single-celled like ancestor into a whale. So my whole point to wrap this up is I don't believe evolution is falsified, because every time a line of evidence comes about that poses a major problem to evolutionary theory, what they do is they change the story. Oh, well, we've got low genetic diversity in humans today, so there must have been some type of hypothetical out of Africa bottleneck, even though that bottleneck would have been extremely damaging genetically. So it's not feasible. So I just think it's circular. They're always going to force fit the data somewhere. They're going to adapt the story. And no, I don't think it's falsifiable. That's my answer, Praise.
Awesome, SFT. Do you want to make a quick response to that, Jackson? Go ahead.
Yeah, I don't think the young Earth creation model is really falsifiable, because I mean, just look at the ICR and AIG statements of faith that I'm not equating you with them. I mean, I know you probably have more integrity than they do, but they say in their statement of faith that no amount of evidence would ever change their mind. And evolutionary theory does change with new evidence. And if evidence came about that totally ruined it, I think everyone would accept that it was done. But I don't think that's going to happen. There's a lot of evidence in support of it.
Great. Appreciate your answer. So we got to go to our next super chat. Steadfast and easy for $5. Thank you for your super chat. He says, or he or she says, "Ha! Bet you thought you lost me. Hee hee. Hi, fellas. God bless."
God bless you as well, brother, and thank you so much for all the amazing support from the chat. This debate's been a lot of fun, and we've had a very lively chat and a lot of good people. And Jackson, you've been a lot of fun to debate. So thanks for the super chat, guys.
Yeah, it's been fun.
Yes, it's been pleasurable to moderate as well. So here now we're into the Q&As, and I mean the questions instead of the super chats. Now we're just in the general questions. Now Landon Freeman, he says, for a question for you, Jackson. How do organisms acquire the intelligence needed to know how to use newly evolved features?
Well, I would say the brains evolve with the bodies. It's a gradual process. I'm not a biologist. I can't really answer it in detail. But the more neurologic function you have, the more your neurons are going to develop. And again, not a biologist, but that's basically my answer.
SOT, if you want to respond to that, go ahead.
Yes. Can you repeat the question real quick?
How do organisms acquire the intelligence needed to know how to use newly evolved features?
Yeah, I would say to begin with, what we see in terms of the forward-thinking mechanisms in our genome, right? Epigenetics, these amazing control systems, okay, and the genomes of living organisms. We literally have millions and millions of switches just waiting to be turned on via environment. Okay, rapid adaptation is what we see. So here's the thing, is evolution doesn't have a mind. Natural selection sees the short term, okay? Natural selection wants the organism to survive till tomorrow. It doesn't worry about getting cancer 50 years from now, okay? So natural selection doesn't have a mind. So what type of evolutionary processes can build that which requires a mind? And if evolutionists want to say epigenetics, for example, is the result of evolutionary mechanisms, then they are applying a mind to evolution, a mind to nature. And also what we have is evidence for interdependent and obviously engineered, simultaneously assembled body parts that could not come together without intelligence. And these are some slides from Genesis Apologetics, as Dr. Dan Biddle has been such a blessing, and he's given me permission to go through a lot of his slides because he does some great work. And he points out here the 5 different parts of our hearing systems work in coordination and how incredible it is. Sound waves are captured by the outer ear, which in turn wiggle 3 tiny little bones called the hammer, anvil, and stirrup, which press on the cochlea, turning into the mechanical process into a hydraulic one. Then you can just go through all of this and how all of this has to come about at once. It can't happen through slow and gradual processes. Okay, what we see in terms of the amazing design of living organisms, God said he created things very good. They had to start that way at the beginning. And of course, degeneration has been introduced since the fall, but things started off perfect. And these things had to start off perfect and perfectly designed and not built up through evolutionary processes. I yield.
Awesome, SFT. If Jackson, if you want to make a quick response to that, go ahead, man.
Yeah, that's the irreducible complexity argument in the Dover trial, I believe it was. I forget what the full name was, but Michael Behe argued that, and biologists countered with, well, a mouse trap may not be functional if you take it in its intended purpose, if you take a part out of it, but he gave an example. Well, with the spring, it can still be a tie clip. You know, these different things can serve different functions. They're not necessarily built with the goal in mind, the end result. And individual components can still have some function without the whole system in place at once. So that's my answer.
Great. And we'll go to the next question from David Neff. Coming at you, SOT. I have a question for both. Sorry.
He's coming at us both then.
Yeah. That was a facepalm moment. Okay. So what evidence would change your mind? So I guess he's talking about creation for you, SOT, and evolution for you, Jackson.
You can go first if you want, SFT.
Well, I guess we've kind of answered that question earlier.
Yeah, we kind of did.
And what I pointed to, I guess I could point to a number of things, but the biggest thing would be testable predictions. What's the gold standard of science? The gold standard of science is to make testable predictions, future testable predictions. So whoever is making the best testable predictions will essentially win the war, okay? Creation versus evolution. And what we have now in 2021 is genetics, where genetics is the best way to determine ancestry. Because as I've pointed out, fossils are not inherited, sperm and egg, and oftentimes you get contradictions in the fossil record. Things in the fossil record are always being overturned, okay? We see stasis in the fossil record, and we see the fact that convergent evolution is a real problem for universal common ancestry. So if it comes down to genetics and the empirical data, who's making the best testable predictions? We've got Dr. Nathaniel Jeanson, who has made testable predictions on the Y chromosome, the mitochondrial DNA. He's making testable predictions on the history of civilization. That, if true, can only be true if young earth creation is true. Okay, he's making testable predictions in terms of mutation rates in people groups where we don't yet know their mutation rate, like the Khoisan peoples. And we have prediction after prediction after prediction on DNA function because we don't assume that what we're looking at in terms of genetics is due to a long history of evolutionary processes. So we don't expect evolutionary leftovers in genomic fossils. Of course, we expect mutations and some degeneration, but not nearly as much as the evolutionists would expect or predict. So we're the ones as young Earth creationists that are making the testable predictions. But if the evolutionists could make better testable predictions that are shown to be successful, then that would be another line of evidence that might convince me that universal ancestry is slightly more plausible than what I see it as now. I know that was long-winded, but yeah, go ahead, Jackson.
All right. Well, what would convince me that evolution is wrong and deep time is wrong? You got to convincingly disprove radiometric dating, because there are many instances where it does work and it's been cross-referenced. But you got to show ice cores are all wrong because they go back 55,000 years, the annual rings, before it gets too compacted to read. But then further dating methods dating to 800,000 years. You got to disprove that there's a glass sponge that was found to be 10,000 years old, older than the Earth is supposed to be. You have to disprove paleomagnetism. You have to disprove sediment pores. You have to disprove just all kinds of things. And but what would convince, if all those were overturned and something was truly found to be thought to be old but proven to be young, that would convince me. And if a transitional fossil, where a fossil was found in the wrong place, say a T-Rex was found in the Cambrian, and a Nomalocaris from the Cambrian was found in the Permian, then I would seriously question these time scales and this the whole evolutionary theory. So that's my response.
Gotcha. Thank you for your answer. So you have a question from Health first, and Filling second. Question for you, Jackson. Why, despite supposedly sharing 99% of our protein coding genes with chimps, do we look so incredibly different?
Well, 99% is a lot, but 1% of what is it, 3 billion or 4 billion base pairs is also a lot. So there's going to be some differences, inevitably, but there are a lot of similarities too, genetically and anatomically. So that's my answer.
Okay.
Please, if I could, yeah, I'll respond.
Yeah, go
real
ahead,
quick.
SFT.
What's also problematic too is the fact that the evolutionists will oftentimes say, we're 97, 98, 99% similar genetically to chimpanzees. But it turns out that, for one, less than 2% of our genomes are protein coding. And many, many genetic sequences are not even comparable between humans and chimpanzees. And there are entire gene families that are known in humans and are not even found in chimpanzees. Okay, so when we consider overall size differences, architecture, gaps, copy number variations, a lot of these things that the evolutionists have ignored, we find roughly 400 million DNA differences, single letter differences that separate humans and chimpanzees that, guess what, have to come about, they have to arise and then spread through the populations to the point of what? Fixation. Okay, we've also got, and I'll end it here, we've also got tens of thousands of chromosomal rearrangements that also had to arise, spread, and become fixated as well. Rates of fixation are extremely slow, and most new variants are lost, of course, because they are rare. So the point is, there's a huge waiting time problem to explain how these vast amounts of differences can arise in just a mere 6 million years. Evolution alone has been falsified because of these numbers. The waiting time problem, a new paper came out recently by Dr. Sanford in a secular journal. It has not been answered. It's been read by 10,000 scientists and it's solid, and there's been no real rebuttal other than a couple, YouTube videos or angry bloggers. But I mean, no one takes those seriously. So that's a huge problem for evolutionary theory and ape to man evolution. I yield.
Thank you for your statement there. But Jackson, you want to quickly respond to that? Go ahead, before we go to the next question, because it was addressed to you.
Yeah. What was the question again? I'm sorry.
Let's see here. Why, despite supposedly sharing 99% of our protein coding genes with chimps, do we look so incredibly different?
Oh yeah, I answered that already.
All right, well, so we'll go to the next one. So
Yeah.
this is from CJ Cox, aka the Synagogue. He has a question for you, Jackson.
Okay.
How can humans and Neanderthals mate and bring forth viable offspring if they are not the same species?
Well, there are many cases of hybridization between different species that produce fertile offspring. And like I said, we differ by 202 base pairs, so we are a different species. But Neanderthals were either subsumed into our genome through breeding, or we wiped them out, basically. So that's my answer.
And SFT, if you want to go ahead and respond to that.
Yeah. If I could, to answer the Neanderthal question is, and I'm just looking for some slides here. As Ken Hovind says, it's nice if you ask the questions in the order that I have my slides, then they're easier to find. When you got a million slides, it's difficult. But I found some slides here. So here's the problem. There has been a drastic revision in Neanderthalensis, okay? In 1856, and this is why evolution is harmful, okay? Because in 1856, this is how they depicted Neanderthal. 2015, this is a digital reconstruction. I mean, it's obviously a human. And what we have in terms of evidence, okay, is that they buried their dead, used medicine, okay? They had complex hunting patterns, made complex tools. They dived for shellfish. What we see, once again, just like with Erectus, we see purposeful navigation, okay? And we see that since we have their genes and we actually interbred with them, and the fact that humans and Neanderthals are less different than polar bears and brown bears, obviously they're humans. They're 99.7% similar to humans. Now, of course, that we'd expect them to be different because they are ancient man, they were earlier, and they also, as I showed in a number of sources, they were highly inbred, 40% less fit than modern humans. This is the end stage of the Neanderthal, where they accumulated so many deleterious mutations, okay, a lot of them coming to fixation, which is why their mitochondrial DNA and nuclear DNA is slightly different than modern humans, but it's because they experienced different environmental conditions. They had a different history. They were the most inbred people on the planet. Their runs of homozygosity are fascinating. So of course they are going to be different, but a lot of the features that we see, okay, we see in people groups today. For example, here, the skull cap, if you found that isolated, the evolutionists would conclude, because oftentimes we find these fossils and these bones in isolation and in fragments. And so they can find a skull cap isolated and they can conclude this might be from a Neanderthal, even though these same features exist in humans today. I yield.
Very good, SFT. So Jackson was addressed to you. If you want to say something really quick, go ahead.
Well, I'm not arguing that Neanderthals weren't human-like. They evidently had culture. They had maybe even religion, and they did bury their dead. They had probably instruments. They made fire. But and if they were alive today, we would definitely give them human rights, and as we should. But they weren't genetically human, and I think that's been proven conclusively. And I yield.
All right, so here is our very last question. It's addressed to you, Jackson. So here's what he asked. So, what about where her spine enters the skull? So
The
whatever
foramen,
that means, I don't know.
The foramen magnum. Yeah, it enters. It's kind of intermediate with afarensis, but other Australopithecines have it more forward than backwards. But the shape of the spine also indicates that it was bipedal. It's kind of an S shape like ours, instead of an arch shape like a chimp. So it definitely walked upright, at least most of the time. it was still capable of climbing, evidenced by its arm structure, but it was a biped, and I yield with that.
I'll respond to that too, Praise. So actually, I'll see if I can find the. Okay, so when it comes to and I showed some slides earlier on Lucy and the foramen magnum, and like I said, I got so many slides here. So, but I do have notes based on what he said in his opening. And the fact is, Lucy's foramen magnum lies in much the same position as some apes do today. So to claim that Lucy walked upright is actually misleading. And the best case scenario is probably that Lucy might have had a bit of a unique walk, but not a walk similar in terms of humans where we walk upright. And when we actually, here's something that's interesting, when we actually account for size differences, okay, with Lucy, then a living gorilla and an Australopithecine, its foramen magnum would be nearly identical. Because remember, these Australopithecines, Lucy, for example, was 3 or 4 feet tall. Okay, so a lot of it is based on misinterpretations and erroneous reconstructions because, as I pointed out, we have these isolated bones where oftentimes sexual dimorphism is invoked, as I discussed in my rebuttal. And so you've got the terrestrialists and the arborealists that are emphasizing different bones. And a lot of times those bones they're emphasizing are actually human bones. And so, of course, if you have a human bone that you're misclassifying in the afarensis category, remember, Lee Berger admitted that afarensis is a chimeric species, then, of course, you can look at a human bone and say, look at this. Lucy was kind of human-like. No, that's a human bone. And this is real. You know, creationists aren't making this up. This is in the literature. Paleoanthropologists admit that oftentimes, I mean, it was Ian Tattersall that admitted that habilis is kind of a wastebasket of bones. So I'll yield there.
Well, that concludes the debate, guys. Awesome job.
All right.
Great questions and answers. Super chats. Well, I'm going to have to give a quick shout out to Logical Plausible. Probable. He's having an after show. So head on over there. You're welcome to head over there, both SFT and yeah. So if you guys, and Jackson, if you guys want to do that, go ahead. But I'll let SFT conclude it.
Awesome. Well, we're going on 2 hours and 15 minutes. We still got over 50 people in the chat. It's been a lot of fun. This is a great topic, great, respectful, cordial debate with Jackson. Yeah. I think we touched on a number of awesome points. The Q&A was awesome as well. Tons of good questions that we could go back and forth on. And yeah, thanks so much, Jackson. And hopefully we can do this again. Jackson,
thank you.
Thanks for doing this. Any final words from you, Jackson?
Yeah, I just want to say you're going to have to send me the link to the after show. I'm not going to know how to get there. I'm terrible with technology.
No problem.
And but also, yeah, again, thanks for having me, and thanks for being respectful. And I respect you a lot. You're very knowledgeable. So I hope we can do this again. It was honestly a lot of fun. I thought I would be really nervous, and honestly, I was for like 30 seconds, and then it faded away. And so, yeah, it was fun.
That's usually the case. Leading up to the debate, and then the first few minutes where you're kind of getting into your opening statement, that's where typically you're the most nervous. But then you kind of warm up, especially in a cordial discussion like this. And I'm glad that you brought up some good points and some good arguments that we could touch on and discuss. So I appreciate you making this a fun debate and an engaging debate. And Logical, Plausible, Probable, his after show. Jackson, I'll send you the link because you are invited, and it'll be similar to the link that I sent you here. It's a StreamYard link, and there should be a bunch of people in there and continue the discussion.
All right, yeah, just email it to me and I'll join.
Okay. Well, Praise, thank you so much for moderating. You are the award-winning moderator, just like George is the award-winning co-host. So great job. You moderated a great debate last week with Bill Tavis. You moderated a great debate tonight. So you've got a really strong career in moderating ahead of you, Praise.
Appreciate that. Thank you so much.
All right, guys. SFT is out.